textbook, you can find a lot of this information in chapter 15. We are going to start by talking about embryonic development. Now, we're talking about the same structure the whole time, but it's typical for people to refer to the developing human as an embryo for the first 8 weeks and then as a fetus thereafter, but it hasn't changed. It's the same structure, it's just gotten more developed. Now we need to talk about what fertilization is. Now we've seen this before, way back in chapter 16 when we were talking about life cycles of organisms. Fertilization is when a sperm are haploid, so N, penetrates an egg, which is also haploid. And the nuclei combine. So at this point, we have a structure that has 2 of every chromosome and not just 1. Now, if we're talking about a human, which we are, this initial structure, zygote, and it has 46 chromosomes. Now, if we were talking about a different species, 46 would not be the diploid number of chromosomes, but we are talking about humans, so 46 is the number. Now, one of the things you need to understand is where this all might be taking place. So, this is the female reproductive system. It's similar to the picture that we've seen before. Sperm would start their journey through the female reproductive system by passing through the vagina and then the cervix. Then through the uterus. And then they have a choice. Sperm are not particularly smart. They don't know where the egg is located. On this particular picture, perhaps you can see right here, there is the egg. So this is the side where ovulation is happening. Some of the sperm are not going to make it to the egg. They're going to go the wrong way. A lot of times the sperm think that the uterine wall is what they should be penetrating, and they bang their head against that. About half of them that make it through the vagina and the cervix will go down the wrong fallopian tube. So they don't know where they're going. They're not really sure what they're looking for. But a whole bunch of sperm are not going to make it. If sperm actually make it down the fallopian tube, they will meet up with the egg right about there. Not inside the ovary, because the egg hasn't been ovulated yet, and not inside the uterus, because the egg can't get there that fast, but rather in the beginning of the fallopian tube. So this point right here, this is where fertilization happens. So this is our fallopian tube, and this is the ovary. Now that's what happens, and that's where it happens, but you also need to know how it happens. So there's this other picture down here to give you an idea of if we were to zoom in on the outer layer of the egg and on the head of the sperm, what's happening. Down here we can see that the sperm acrosome, which was a part of the structure of the sperm that we labeled previously, the acrosome is the part that has enzymes that are built to digest the outer layer of the egg. But the acrosome digests and then helps to penetrate the outer layer of the egg. Now, the outer layer of the egg is referred to as the corona, and zona pellucida, which are just outer layers of the egg. So if you can see what's happening in the picture, here's the acrosome right here. You can see the contents of the acrosome spilling out, all those little dots. Those are the enzymes that we said were in the acrosome. They help break through and penetrate the outer layers of the egg so that eventually the nucleus of the sperm, which on that picture is the purple piece, can combine with the egg. Now, what happens if we think about other things that we've learned about before? When the sperm penetrates the outer layer of the egg, it depolarizes the egg membrane, and this depolarization prevents other sperm from entering. And if you look at what's going on, it's not the entire sperm that's going to go inside of the egg, but rather the sperm head helped digest the outer layer. And if you look, just this purple chunk, which I said was the nucleus, that's the only thing that's actually going inside of the egg. So we would say only DNA or the nucleus enters. The rest of the body of the sperm, the part that had the mitochondrial spiral in the body and the tail for movement, those parts do not enter. Just the DNA does. So when you did your lab, and if you look on Google Classroom, I posted the marks to those yesterday, and you can see if I put any comments, maybe you're missing something, but most people correctly talked about the fact that the sperm needs to be small because it needs to transport itself, and it takes a lot of energy. If it was bigger, it would take more energy. There need to be lots of them because a whole bunch of them are going to get lost on their epic journey that they're going on. And the sperm need to have that head with the acrosome on it so it can digest the outer layer. Sperm are only contributing DNA to the developing embryo, whereas the egg is contributing the rest of the cytoplasmic contents, which is one of the reasons that the egg is so much bigger than the sperm. The egg doesn't need to move, not by itself anyways, so it doesn't require a tail like the sperm do. But those are some of the things that were good explanations for the differences in size and in number between egg and sperm cell. Anyone want to ask anything there? Now, once the sperm fertilizes the egg, we start a new time count system. Prior to that, we might have been thinking about, in the female's reproductive system, what day of her menstrual cycle is she on? If the sperm is fertilized, or if the sperm fertilizes the egg, now we can start counting how many days or weeks old is that zygote, which will become an embryo. So if we look at this picture, here is the ovary. We can see that the ovary has been at work and an egg has been ovulated. And like I mentioned earlier today, you have about 24 hours or so for the egg to be fertilized. Once that happens, once the egg is fertilized, then things continue to change. Now, if you see right here, there are a whole bunch of sperm surrounding that egg, but only one appears to have penetrated it. So here is our zygote. There is where fertilization took place. Now, if we were to take time-lapse pictures, so this is not happening at the same time, this would be over time, what happens to the zygote. It's going to be moved along the fallopian tube. The egg itself isn't mobile, but the fallopian tube will help wash the fertilized egg or push the fertilized egg down the fallopian tube. Now, if we think about what is happening from this point until it gets to the end of the fallopian tube, we started out with 46 chromosomes, one cell that had 46 chromosomes. This arrow here represents segmentation. Sometimes the word cleavage gets used there. But what we mean by segmentation is really that mitosis is happening. Mitosis will occur, and we will continuously double the cell number as time passes. So I started with one cell, then I would have 2 cells, then I would have 4 cells, then I would have 8 cells. So we continue doubling the number of cells through the process of mitosis. Now, when we get over here to about the 7th day, the structure that we have is called the morula. This is when there are 16 cells. So the egg was released. Within 24 hours, it got fertilized. And by the time about 4 days have passed, that one egg got fertilized, underwent mitosis, and traveled the length of the fallopian tube until it was just entering into the uterus. Now, once it enters the uterus, we get a different looking shape. And I'm just going to draw it again a little bit bigger so that you can see what it looks like. Until that point, it just looked like a clump of cells. There was 1, then there were 2, then there were 4, then there were 8, then there were 16. Starting at around the 4th and a half or 5th day, so after it makes it to the uterus, it starts looking a little different. There will be this section here, the inner cell mass. So there'll be a clump of cells that look like they're clustered on one side of this structure. The inner cell mass, that's what's going to become the embryo eventually. So all of those cells that we're duplicating, a bunch of them clump on the side. Then there's a bit of a cavity there. And then we have more cells that form like the outer ring of things. This is called the trophoblast, and it will become first something called the chorion, then the placenta. Now we're going to talk about the chorion and the placenta a little bit later. But essentially, we've divided this clump of cells into 2 things. There's a clump of cells on the side that will eventually become the embryo, the baby. There's a space. And then there is this layer of cells along the outside. The chorion and the placenta are both structures that are going to support the embryo. So there's lots of support structures that are involved. And notice they all came from that same egg that was fertilized by a sperm. So all of these cells have the same DNA, but they're getting different functions. This structure is called a blastocyst. Now, like I said, this is on around day 4 or 5. So after it has made it to the uterus, but before the next thing happens. Right here, around 6 days, so a week or so, that is when implantation occurs. So we're looking at just the side of the uterus here. The best place for implantation is right there. In the endometrium, the nice thick layer of the uterus. It's thick because estrogen and progesterone were being produced and we're making it thick. If we compare these days to a woman's cycle, so I'm just going to put some green numbers here. Day 14 is when all this stuff happened over here on the side. She ovulated, the sperm fertilized the egg within a day of that happening. Then about a week has passed. So around day 20 or 21, in the woman's cycle, that is when, if the egg were fertilized, it would be implanted. So if we compare this, what's going on with the embryo and what's going on with the woman, she ovulated, about a week passed, embryo implanted in the uterus. Do you think she would know she's pregnant at this point? No, and she probably wouldn't guess that she was unless she was expecting to be, because on day 21, she's not expecting to have her period yet. She wouldn't be expecting to have her period for a whole another week after this, and we'll see that a whole bunch of things will happen in that week of time between when implantation occurred and when she might clue in that she's pregnant. At this point, it would be tough to detect pregnancy on day 7. There are some pregnancy tests that could potentially detect it, but they're looking for a level of a hormone being produced. And at this point, it would be really low. It would be hard to detect. Not impossible, but hard. Now, what happens next? Yeah. What do you mean, how do people get pregnant later on? So the difference would be, here, I'll just go back to this one. Remember I said that day 14 is like what would happen if the cycle were 100% perfect? Some people are not ovulating on exactly day 14. Some people have longer cycles, some people have shorter cycles. Some people ovulate farther into the second half of their cycle than, or farther or closer to the first half of their cycle. So if a person's cycle was shorter than 28 days, and ovulation was a little bit early, she might still be having her period when she's ovulating. So because these are just what would happen if everything was perfect numbers, remember that they don't always line up with reality. Technically, regardless, you really only have 24 hours from whenever the egg leaves the ovary before it's fertilized. Otherwise, it's not going to survive. Hello. It's really a miracle that anyone has ever had a child ever, because all of these crazy things have to happen for it to work. Okay. Okay. Well, then I'll just keep talking about babies. Does anyone else want to ask anything on this slide before I go ahead to the next one? Okay. So the next one, and you have this picture, because I don't know about you, but some of these structures are pretty sketchy for even me to draw, and I know what they are. Gastrulation is the thing that comes next. This process happens during the second week. So when I say the second week, I mean the second week from when the egg was fertilized. The first week was consumed with the egg going through mitosis, making its way to the uterus and being implanted. Once it's implanted during the second week, that's when gastrulation happens. Gastrulation is where differentiation starts. Now, until this point, remember, we just had cells. They don't really appear any different. They might have moved to a different location in the embryo, but their structure is still the same. During the second week, that is when the cells start determining what kind of cell they're going to be. So differentiation means cells becoming more specific in function. So we had a sperm fertilize an egg, then we had a cell, then we had 2, then we had 4, then we had 8. Now we have this big clump of cells that are actually going to start becoming different types of cells, having specific functions. So we take our blastocyst, and you can see the word here, blastocyst, and the cells start migrating. So if I consider my blastocyst over here, I have this clump of cells that's on the side, right? Clump of cells right here. And it says it starts migrating inward. So basically what we're saying in gastrulation is that that clump of cells folds in on itself. You would not ever have to label a diagram of this process happening, but I wanted you to see what it looked like because I find that really hard to describe with words. So if you follow these arrows here, the cells that are in that inner cell mass fold in on themselves. The result is that you get 3 different layers of tissue: the ectoderm, the endoderm, and the mesoderm, which are the words that are on your handout following this. So what happens during gastrulation? The blastocyst cells fold to form 3 germ layers. And here they are, the 3 germ layers: ectoderm, endoderm, and mesoderm. Notice how there's still part of that clump of cells that's on the side, and there's a big cavity still on one side of our blastocyst. Now the last thing I want us to look at today is these 3 germ layers. These 3 layers will each become a different type of cell in the developing embryo. And so the thing I'm asking you to do is to take a look using your textbook on page 513 at which types of tissue the ectoderm, the mesoderm, and the endoderm will become.